Okay, today I'm going to talk about the atomic absorption lab, and specifically how to prepare your standard solutions of calcium so that you can determine the concentrations of calcium in your two unknowns. In the next video, I'll talk about how to prepare your calibration solutions for the magnesium. So for the concentrations, the purpose of this lab is to determine the calcium and magnesium concentrations in two unknown solutions. The water hardness unknown solution from your last lab experiment, and a solution of tap water that you're going to bring from home. So you're going to work in pairs for this lab. So both you and your partner need to analyze each of your water hardness unknowns and tap water samples for both calcium and magnesium. To get data from the atomic absorption spectrometer, you're going to need to prepare a set of calibration standards that are going to allow you to determine what the concentration is in your unknown solutions. So you're going to analyze a set of solutions that you know the concentrations of and get an instrument response. Using those two variables, you're going to construct a least squares linear regression line. And then you're going to use that equation when you analyze the solution of unknown concentration to use the response to calculate the concentration. So the first thing you're going to do is prepare your calibration standards of calcium. So you probably prepared a solution of calcium carbonate to standardize your EDTA solution, and that's the solution that you're going to use to prepare your calcium standard solutions for this experiment. So your calcium carbonate solution probably has something around 0.015 molar calcium carbonate. So the first thing we're going to need to do is convert calcium carbonate in molarity to calcium in molarity. So if we have 0.015 moles calcium carbonate per liter calcium carbonate, then we can use the ratio, the stoichiometric ratio, to convert that to moles calcium. So now we see that that's equal to 0.015 mole calcium 2 plus per liter. Okay. The next thing we're going to need to do is use the formula weight of calcium 2 plus to convert the molarity of calcium to parts per million concentration units. The important part to remember here is that even though the stoichiometric ratio makes it so that the molarity of calcium carbonate is equal to the molarity of calcium, that's because that's a mole-based unit. When we do parts per million, when we use parts per million units, that's a mass-based concentration unit, and so the concentration of calcium carbonate is not equal to the concentration of calcium. So to convert from molarity to parts per million, we're going to say that 0.015 moles calcium per liter. We're going to multiply that by the formula weight of calcium, 40.08 grams per mole. And that's going to give us 0.6012 grams calcium 2 plus per liter. Now, the way that you would do this calculation, move to the other side of the board here, the way that you do this calculation, the long way is to kind of use the density of water to convert between grams and liters and figure out what how many parts per million this is. A shorthand way to do this is for aqueous solutions is to understand that 1 milligram per liter is equal to 1 ppm. And so when we understand that, then we can see that this number is really equal to 601.2 milligrams per liter of calcium, which is equal to 601.2 ppm of calcium. So that is the concentration of the standard solution, and now we're going to make dilutions of that standard solution for our calibration curve. And I'm going to do that in the next video.